Welcome back friends in another video tutorial for microbiology with Shagnik. So in this video tutorial we will going to discuss about the next part of our pentose phosphate pathway, that is the non-oxidative phase. In the previous video we have already talked about the oxidative phase. So in the previous video we have discussed the oxidative phase where we have seen the end product was ribulose 5-phosphate. Okay, so from this ribulose 5-phosphate, we will going to do our discussion today. Okay, so if you have not watched the oxidative phase and you don't know about the oxidative phase, then I want to recommend you to go and watch that video first. Then it will be helpful for you to understand the thing that I will going to teach you today. Okay, so let's get started with the video. So first thing that we should remember, that is in case of non-oxidative phase, wait a minute, that is in case of non-oxidative phase, three molecule of ribulose 5-phosphate will going to enter. Okay, and interconversion of this three ribulose 5-phosphate will occur. Okay, that ultimately gives us two 6 carbon containing glucose 6-phosphate and one 3 carbon containing glyceraldehyde 3-phosphate. What is the meaning of interconversion? Interconversion means there will be transfer of carbon containing molecules. That may be 2 carbon containing molecules, that may be 3 carbon containing molecules. That how various type of interconversion will occur. Okay, so let's see how these interconversions are going on, that ultimately giving us the end product of pentose phosphate pathway. So first, I have taken this picture from CSIR Pathfinder Life Science book, volume 1. So you may also refer this book to understand the thing. So now let's try to understand the entire thing. So first of all, the main reaction of non-oxidative phase is mentioned here. That is 3 molecule of ribulose 5-phosphate is converted into 2 molecule of glucose 6-phosphate and 1 molecule of glyceraldehyde 3-phosphate. Okay, so now 3 ribulose 5-phosphate containing molecule are entering, right? So first what will happen, that 1 ribulose 5-phosphate will be converted into xylulose 5-phosphate. Now before going to talk about this reaction, we should know some basic thing about carbohydrate. That is, the basic unit of carbohydrate is known as monosaccharides, right? We all know. Now monosaccharide, on the basis of the functional group it containing, they can be divided into two things. One is ketose, another one is aldose. Ketose means monosaccharide containing ketone group, and aldose means monosaccharide containing aldehyde group, right? So now whenever you see any kind of ketose, make sure you will find something eulose in its name. Okay, but you will not going to find this kind of thing when you are try to reading any kind of aldose. Okay, so what I am telling this to you, that ribulose 5-phosphate, you are finding something like eulose here, right? And xylulose 5-phosphate, xylulose, right? So eulose. Okay, so that means both ribulose 5-phosphate and xylulose 5-phosphate are ketones, right? They are having ketone group. Okay, so here the change is only in one carbon atom. Okay, and just a simple change, that means suppose if in the structure of ribulose 5-phosphate, one carbon contains H in the left side, another one in another OH in the right side, then in xylulose 5-phosphate it will be changed in such a way. Okay, and this kind of change is known as epimeric change. Okay, that means ribulose 5-phosphate and xylulose 5-phosphate are epimer of each other. Okay, so now this kind of change will be facilitated by enzyme known as epimerase. Okay, so this is the first enzyme that will going to take part in non-oxidative phase. Now in the second reaction, ribulose 5-phosphate will be converted into ribose 5-phosphate, right? There is no eulose type of thing in case of ribose 5-phosphate, right? That means it is not a ketose. This thing is very very clear, right? So it is a aldose. Okay, so as it is a aldose, that means ketone group is transferring into aldehyde, right? that means there is a isomeric change, right? So ribulose 5-phosphate conversion into ribose 5-phosphate is facilitated by an isomerase, that is phosphopentose isomerase. Okay, and the third molecule of ribulose 5-phosphate is converted into xylulose 5-phosphate, obviously with the help of epimerase. Now, for learning the remaining part of non-oxidative phase, we should know about the function of transketolase and transaldolase. So let's talk about them first. So transketolase. So remember, in case of transketolase, its main goal, as its prefix suggests, is to transfer. Okay, so now transketolase will going to transfer two carbon containing molecule. Okay, now the question that will arise, that is who will going to be the donor and who will going to be the acceptor. So donor will be a ketone containing monosaccharide ketose, and acceptor will be a aldehyde containing monosaccharide aldose. Okay, in case of transaldolase, same similar thing will going to occur, but it will transfer 3 carbon containing molecule. Okay, I hope you can understand. So in case of transketolase, it is 2 carbon containing molecule, and in case of transaldolase, it will going to transfer 3 carbon containing molecule. Okay, now another thing that you have to remember, that is in case of non-oxidative phase, remember ketose will going to play the role of donor. Okay, as we have just seen, and by transferring the thing, it will become a aldose. Okay, this something will going to help you to understand and remember the entire non-oxidative phase. Now let's try to test the concept we have learned just now. So first is xylulose 5-phosphate. Transketolase will come. Xylulose 5-phosphate, as it is a ketose, it will going to take the role of donor, right? So as it is a ketose and transketolase is here, it will going to donate 2 carbon to its aldose friend, right? That is ribose 5-phosphate, right? Now as aldose is getting 2 carbon containing molecule, that means it itself becomes a monosaccharide of 7 carbon, right? So now, as I have told you, that xylulose 5-phosphate by donating will become into a aldolase, not aldolase, it is aldose. Aldolase is an example of enzyme, and we are here talking about monosaccharide. So it will convert it into 3 carbon containing aldose, that is glyceraldehyde 3-phosphate. Okay, and ribose 5-phosphate, after getting 2 monosaccharide, will be converted into 7 carbon containing ketose, that is pseudoheptulose 7-phosphate. Okay, so the thing that we have just learned is working, right? So now let's talk about the transaldolase. Transaldolase will going to do what? It will going to take 2 carbon from ketone, right? 2 carbon containing molecule from ketone containing monosaccharides, right? So ketone containing monosaccharide is pseudoheptulose 7-phosphate. So it will going to take not 2 carbon, sorry, it will going to take 3 carbon. So 3 carbon containing monosaccharide. No, sorry, not monosaccharide, 3 carbon containing molecule. So from pseudoheptulose 7-phosphate, transaldolase is taking 3 carbon containing molecule. Okay, and after donating 3 carbon containing molecule, pseudoheptulose will going to convert into a aldose, right? So 4 carbon containing aldose is erythrose 4-phosphate. Okay, and as it is transferring 3 carbon to glyceraldehyde 3-phosphate, after getting this, it becomes a 6 carbon containing ketose, that is fructose 6-phosphate. Understood? That is the thing going on here. Okay, now fructose 6-phosphate will going to convert it into glucose 6-phosphate. Okay, and as it is a isomeric change, obviously here the enzyme is isomerase. Okay, now erythrose 4-phosphate will going to react with xylulose 5-phosphate in presence of transketolase, right? Transketolase will going to take 2 carbon containing molecule from a ketose, right? Here the ketose is xylulose 5-phosphate. So from xylulose 5-phosphate, it will take 2 carbon containing molecule, and after donating 2 carbon containing molecules, xylulose 5-phosphate will become a 3 carbon containing aldose, right? 3 carbon containing aldose, what we all know, that is glyceraldehyde 3-phosphate, right? And erythrose phosphate, after getting 3 carbon from xylulose 5-phosphate, no, sorry, not 3 carbon, 2 carbon from xylulose 5-phosphate, it will convert it into glucose 6-phosphate, right? That is the entire thing. Just remember that transketolase will going to take 2 carbon from any ketose, and transaldolase will going to take 3 carbon from any ketone, right? And after transferring, that ketose monosaccharide will be converted into aldose, and aldose monosaccharide, after getting 2 or 3 carbon containing molecule, converted into ketose, right? Just by understanding this simple concept, you can understood the entire non-oxidative phase. Okay, so that is the idea. Hope you can understand the entire thing. Okay, so another important thing that I should want to mention, that is transketolase catalytic activity requires thiamine pyrophosphate, a coenzyme that involved in various kind of transfer reactions, right? And here transfer reactions are going on, so it is very important here. Okay, so here we have mentioned that transketolase and transaldolase will going to transfer through 2 carbon containing unit and 3 carbon containing unit respectively. In each case, it will going to take that 2 carbon or 3 carbon from ketose donor and transfer it to a aldose donor. Okay, so that is the entire idea of our non-oxidative phase. So I hope this video will be helpful for you for understanding the entire concept of non-oxidative phase. We will again going to meet in our next video, where we will going to talk about function of pentose phosphate pathway. So till then, bye bye.